Hey, greetings from the BrainGate channel. I'm just going to talk a little bit about the timing perception confound. We've actually published a bunch of stuff on this, but not many of you use this measure because you kind of prefer to do a shorter battery. But the, uh, the basically what the timing perception confound is an additional test that we do in addition to timing perception. So normally you do timing perception in your test battery, but timing perception confound gives you an added dimension of plasticity. And it's basically an illusion that makes that increases the sense of timing perception, makes it feel like well, basically it's an illusory confound that goes along with duration discrimination and makes you worse. It makes you worse at that particular test, and but only if there's something wrong. And neuroinflammation impacts that score, reduces the difference between baseline and illusory condition, and that difference is normally about 30%. But if it's neuroinflammation is bad enough, it's going to remove it, and that gives you that interesting problem or that interesting phenomenon where people who have problems actually outperform people who don't. So it gives you a little more insight into that particular group. But timing perception again is asks the question which stimulus lasted longer. Visually, you can have a sort of a length illusion. You draw these two lines here that are the same length. One sort of looks longer than the other, but you know that's an illusory confound. So can we design a test that changes the context of how long a tactile stimulus lasts? And I'm just going to give you a little bit of science just so you have a little bit of a feel for this. And this is the experimental basis for this. This is basically from an optical imaging experiment, or in this case, a combined experiment where we both did optical imaging and electrophysiological recording. In this case, we had an electrode put here, and this was an optical image. And I'll get to what optical images are in a minute. But this dark area is where a stimulus on the finger caused enhanced activity here. And if you deliver a long stimulus, in other words, if this stimulus duration lasts 5 seconds, you see that responsivity from the electrode goes way down. In other words, the brain is getting bored with that stimulus. So, you turn the stimulus off and it sort of goes off, but, there's not a whole lot of action response. That's the main thing to think about. Your responsivity is going down. You get a very different response from the optical image. So the whole point is with the optical image, you see with a short duration stimulus is this black line. Response to the red stimulus, response is longer. Longer stimulus is green, and longer stimulus is blue. You see, these optical responses sort of are like a memory trace. They last a lot longer. And it's a result of glial swelling because the difference image you see here is a result of taking one image and before you deliver that stimulus to the finger, and then taking another and then subtracting the two and looking at the difference. And those differences are on the order of like 0.01%. They're not very big, but you can see them, and they have this long time course. So they're really, you're looking at, in this case, we're in the near infrared range of like 930 nanometers. So you're really not looking at blood flow. You're looking at a lot of other stuff like glial swelling and extracellular potassium. So anyway, it's a different signal, and it gives you a little bit of insight that you deliver at different stimulus durations, and these things last longer. So you can easily tell the stimulus duration between by looking at these lines. So it's a physical property we can see in the brain where we could actually tell the difference from one stimulus to another. Now, here's the next part of that. If we increase the duration or the amplitude, if we compare the duration of that signal of the stimulus that we deliver here, and we compare a small amplitude with a larger amplitude stimulus, we see that number one, you get a larger peak here, But you also see that this stimulus is going to last longer. And so basically, we're kind of saying, well, that stimulus is going to feel like it lasts longer. Now, if you look at the electrical activity to this, you don't see any, and deliver a 100 micron stimulus and a 400 micron stimulus and look at electrophysiology of that, you don't see a difference in how long that stimulus lasts. But this looks like it's lasting longer. So prediction is, go on to the next slide, prediction is basically, if you deliver a larger stimulus amplitude, it will make it feel like the percept of that stimulus lasts longer. And, I guess the analogy is, press your finger with a pencil and you can feel it when it's on there. Hit it with a hammer, it lasts a lot longer, except that involves pain stimuli. So let's not go there. But basically, though, larger stimuli feel like they last longer. That's the whole point. Now, what is the actual test? Again, baseline test, which stimulus lasted longer, track it down, and you've got some kind of threshold. All right, now, illusory condition. We make one of these stimuli a little bit larger. Make the shorter one a little bit larger, and that's going to make it more difficult to tell the difference. More difficult to tell the difference means you do worse. All right, over here, looking at the difference line. Remember, smaller numbers are better. This is in milliseconds. So, normal, we got, 65, 70 millisecond. And, in terms of what the difference Lyman is, and let's just look at this. Here is control and here is control. We add the confound, all of a sudden you're much worse. All right. Now, what happens in another population? In the concussed population, we add the confound and you don't get a difference. In other words, that illusion that makes you about 30% worse does not make people with a neuroinflammatory problem worse. So these people are actually outperforming the healthy controls because they have neuroinflammation. So they've got some kind of problem. That neuroinflammatory problem allows them to do better. And it's got to do, with the fact that neuroinflammation is removing the illusion. Remember, with an illusion, the way to think about it is the brain is what's connecting the dots, and illusions actually, removing the illusory confound actually makes you perform better. So you should respond to the illusion, but basically this actually contributes to the plasticity score if you're doing that test. Anyway, hope that made sense, and thanks for watching. See you next time.